In this video, we'll learn about the phylum Ascomycota, which are also known as the sac fungi. These fungi get their name due to the formation of a sac that contains haploid ascospores. So if we look at this picture here, so first of all, you can see that these are the sac fungi here. And this whole structure, this bowl here, is actually called an ascocarp. Now within this bowl, if we look at this picture, which kind of zooms in on the microscopic parts of the fungus, we can see that within the ascocarp, we have these little sacs. And each sac is called an ascus, and plural is asci. So you can see that it's pointing to multiple mature asci here. And inside each one of these ascuses, or asci, you can see these smaller little structures, these little circles here. And these are the ascospores. The hyphae, so remember the hyphae is the kind of the underground vegetative body of the fungus. So the hyphae are septated. So they are going to look like this picture here, where each cell has its own cell membrane and cell wall. Whereas with aseptate, remember we're looking at just one cell with multiple nuclei. But here we're looking at multiple cells. And each cell only has one nucleus. The Ascomycota can do asexual reproduction as well as sexual reproduction. And during asexual reproduction, kinetiophores release haploid kinetiospores. And these are structures that are extending from the hypha of the mycelium. And so you can see here, this would be the kinetiophore. And then the kinetiospores would be inside here, and then they would get released out into the environment. I want to point out the kinetiophore on this picture as well, even though we talked about it on the last slide. So remember, the kinetiophore is part of the asexual reproduction of Ascomycota. And you can see that here. So the mycelium essentially produces this structure that has haploid spores, kinetiospores, that can then be released here. So these asexual spores are the kinetiospores. Now during sexual reproduction, the hyphae develop into specialized mating types. The quote-unquote male mating type is known as an antheridium, and the female mating type is known as an ascogonium. And Ascomycota can be either homothallic or heterothallic, which just means that these mating types might be associated with within one single fungal organism, or it can occur with two separate fungal organisms that come together in order to reproduce sexually. And just like we talked about with the reproduction of fungi in general, the Ascomycota go through the same basic steps. So first of all, we are going to have plasmagamy, and remember that this is where the cytoplasm gets married. So remember, it's cytoplasm marriage. So what happens during this plasmagamy, you're going to have a cell from the antheridium merge with a cell from the ascogonium. So in this picture here, we'll say that the cell with the red haploid nucleus is the antheridium. And the cell with the blue haploid nucleus is the ascogonium. And so the cell membrane, cell wall, and cytoplasm of these two cells are going to merge to form one structure, but the nuclei are not going to merge yet. So we end up with the structure known as a dikaryon. And remember that this is not a diploid cell because we still have two separate haploid nuclei present. The cell won't be considered diploid until the nuclei merge during karyogamy. So during karyogamy, that dikaryon that was created during plasmagamy, the haploid nuclei are now going to merge and form a diploid nucleus. And this structure is going to be known as a zygote. And I colored this nucleus in purple because blue and red, when mixed together, make purple. But remember that this diploid nucleus is actually going to have a mixture of red and blue chromosomes inside of it. This diploid cell, the zygote, is now going to go through meiosis. And remember, during meiosis, these chromosomes that came from our haploid nuclei are now all together in one diploid nucleus. And as the cell goes through meiosis, those chromosomes are going to get rearranged. And so this zygote is ultimately going to form daughter cells, but the daughter cells are going to have a different genetic makeup from the zygote, and it's also going to be genetically different from the original cells that came together in order to form the dikaryon. So interestingly, these Ascomycota have kind of an additional step in their sexual reproductive cycle. So after the zygote goes through meiosis in order to form these haploid nuclei, the cell then goes through mitosis and cell division. So this one cell here, the zygote, turns into 4 haploid nuclei and ultimately forms 8 haploid ascospores through mitosis. The ascus then releases the ascospores into the environment, where they will hopefully land in a favorable location where they can germinate and start going through mitosis in order to form a new mycelium.